Hey guys. So I know a lot of you guys have like a lot of exams this week, so I guess we'll just get right into it. So we're gonna do polls again. We're basically for this review just going to be going over questions, and if you guys like have any further questions, we can try and answer those. Um, but yeah, I'll give probably like a minute or two for these first three questions, but they're pretty straightforward. Okay, so just gonna give like 20 more seconds. You guys can get your answers in. Okay, I'm gonna go ahead and end the poll. Okay, so for number one, which of these hormones mediate their effects in target cells primarily via changes to gene transcription? So that would be B. And directly from Dr. Cho's slides, he says that thyroid hormone and steroids both are in charge of activation of specific genes, which helps make new proteins. And then for number two, release of which hormone is stimulated by action potentials between the hypothalamus and the pituitary? That would be C, because only the posterior pituitary gland is activated by action potentials. And the posterior pituitary gland is in charge of releasing oxytocin, ADH, or also known as vasopressin. The anterior pituitary gland is activated by the hypothalamus through hormones. So that's why that would be oxytocin, because only the posterior pituitary gland is activated by action potentials. And then for question three, which of these hormones is likely to mediate the slowest and longest downstream effect? That would be C, because so if you think about what these are, and what they do, thyroid hormone is like going to change gene transcription and like cause new proteins to be made. And that's a pretty long process to like activate gene transcription and everything. So that's why three would be C. And does anybody have any questions on these? No? Okay, moving on. So I'll give you guys a couple minutes for answering 4 and 5 too.
Yes.
It's a little ugly, but I tried my best.
Oh my gosh.
Okay, so I'll just give like 10, 15 more seconds. Okay, so number 4, removal of the adrenal cortex with patients with Cushing's disease will likely have what effect on blood cortisol and ACTH levels? So that would be B. And this is a diagram that Dr. Cho showed you guys. But basically, in Cushing's disease, you have a tumor on your pituitary gland. So that causes the anterior pituitary to overproduce ACTH. But if you remove the adrenal cortex down here, you can't produce cortisol. And cortisol has a negative feedback inhibition with the anterior pituitary. So if you get rid of the adrenal cortex and you can't make cortisol, you can't inhibit the anterior pituitary. So it'll just keep producing ACTH. But again, you've gotten rid of this, so you can't make any cortisol. So therefore, your blood cortisol will decrease, but your ACTH levels will increase. And then for number 5, the answer is B. And I included these two diagrams from Dr. Cho's slide as well. So the sympathetic nervous system, which is your short-term stress response, breaks down glycogen stores, but glucocorticoids break down fat and proteins to increase blood glucose, and that's what happens if you're like stressed for long periods of time. And as far as the exam figures, there weren't many from topic 15. It was basically just this one. So I would definitely recommend reviewing this. And if you guys have any questions about this figure, feel free to ask. I'll give like maybe a few seconds for anyone to ask any questions that they want about this figure, and then we'll move on to the next topic.
Hey, I actually have a question about it. Can you hear me okay?
Yeah.
So basically what the figure is saying, it's just saying that during the month of March, because we were talking about like rainy season from like January to July, that March is just the highest, and we're assuming it's because of like competition, and that's the reason why it has higher GC, right? Is that basically the takeaway from that?
Yeah.
Okay.
All right, so I guess we'll move on to the next topic.
All right, I'll start sharing my screen then. Okay, so go ahead and give these questions a try. Topic 16 is just all about muscles. All right, so 30 more seconds and then we'll go over these. Okay, so let's go over the answers. So almost everyone got the correct answer. So what we're looking at here, right? We're zooming in into a myofiber, which is a muscle cell, and then we're zooming in even more. We're looking at the fibers inside of that muscle cell called myofibrils. Once we zoom in even more, we're looking at sarcomeres, and once we zoom in even more, are looking at our thin filament, which is this whole structure here. So our thin filament is made up of these beads, as we can see. They're labeled A here, and these beads are actin. On our beads, of course, we have our myosin binding sites, which is the gray areas. This rope-like structure here is tropomyosin, which blocks the actin myosin binding sites. And then lastly, we have troponin, which are these extra structures here. And like the results show, everyone basically got this right. Troponin binds calcium here. Upon calcium binding, we shift the tropomyosin, allowing those myosin heads to bind and then do the power stroke. So yeah, everyone got it right. And then one thing to note here is that we are looking at skeletal muscle and cardiac muscle. And how we can tell is troponin and tropomyosin are present. Basically, smooth muscle does not have troponin, it doesn't have B, and doesn't have C. So that's something you should go into the exam knowing. But other than that, this question was pretty simple. We'll do this next question here. All right, I'll go over the answers in 30 more seconds. It's looking good. Almost everyone's getting it right here. All right. So this question is a little tricky because the zones we learn in class, they do not exactly match these regions that the figure is referring to. So what we need to do is first look look at what we were taught in class. So this is what we are taught. A bands, obviously, sorry, let's start with sarcomere. Sarcomere obviously is the entire unit. We go from A to A. Okay, so that's simple there. A band you should know that it's the entire length of the myosin. So if we look here, the C region does correspond to the A band. H zone is myosin only, meaning there's no overlap. So if we look here, there is not a segment that corresponds to the H zone, because the H zone would actually be from here to here. That would be myosin only. I band corresponds to actin only, and actin only would be the light region. And as we can see, the figure does not point this out. And then obviously the Z lines are just these dark lines on the end, and then M line will be the line straight down the middle. So what I was trying to point out here is that these zones you learn in class do not match these regions here. Some do, some don't. But anyways, like I said, the A does in fact represent the entire sarcomere. B represents the overlap. And how you can tell this is the overlap between the thin and the thick is because it's the darkest region. C represents the A band because that's the entire length of the myosin. And then D here represents the entire length of the actin, but it is not the I band because the I band is actin only. So just be sure to understand that this figure doesn't correspond to A band, H zone, I band, Z line, or these bands here. Okay, so they are a little bit different. But anyways, so the correct answer is A and B for these two questions. So as we can see in figure 4, the only thing that shortens is the sarcomere, because once the myosin, which lays right here, starts performing its power strokes, it is tugging on the thin filament. It's basically pulling the thin filament towards the center from both sides. So that's why the sarcomere will get smaller there. And then obviously A band and the entire length of the actin, those do not change. Those filaments do not change length. The only thing that changes length or size would be the sarcomere and the amount of overlap. So as we can see, what gets longer during contraction, it would be the overlap, meaning this gray region would extend farther out as there is more overlap between the thick and the thin filaments. Someone asked, would this muscle be almost fully contracted? I would say yes, it's almost fully contracted, and how you can tell is because the thin filament reaches all the way up to this point. And the only section that has only the only section of thick filament where there is still a little bit left you can pull would be this tiny region here. So yes, this is basically almost an entirely contracted sarcomere. And then we'll do these two questions now. All right, 30 more seconds. All right, so let's go over the answer here. So the majority of you guys got this right. So let's talk about why. So the first step in answering this question is understanding what each of these graphs are referring to. They look similar, but they look different at the same time. So this is telling us that we have a muscle contraction, and then both of these graphs have muscle contractions, but the action potential is what differs. So what we can assume here is we see a plateau here, and what that tells us is that while this is for a regular skeletal muscle, you have one twitch, you have muscle contraction that follows. This plateau phase is characteristic of cardiac cells. You do not see this plateau phase in skeletal muscle, so that's how you can tell that this graph is for cardiac and this graph is for skeletal. And now next we look at this graph here, and what we notice is it's an action potential, but we also see this slow rise here. And so what we can learn from that, we can determine this is a pacemaker cell specifically. And so now let's go over the question. In figure 14, which phase is driven by funny sodium channels? So first off, recall from lecture that funny sodium channels are in pacemaker cells. That's what allows your heart to beat without any nervous input. Remember in the SA node, you have these pacemaker cells that constantly signal the heart to contract. And more specifically, we have these funny sodium channels that are funny basically means they're leaky sodium channels. And so more specifically, during a resting, in a normal cell, what you have is you have leaky potassium channels. What that means is you're always going to be, your cell is going to be always negative unless it's depolarized. The reason why it's called funny sodium channels is because they differ from these leaky potassium channels. They're funny in that usually we're pumping sodium out of our cells, but in this case, we're letting sodium slowly leak into our cell. But obviously, it's important because this slow leak up allows us to reach our voltage threshold, which then allows us to go through our action potential. So that's what a funny sodium channel is. It's just a leaky sodium channel that's always open and allows your cell to slowly depolarize. And so, yeah, so this here, this slow rise here is driven by our funny sodium channels. Now we look at question 18, which phase functions to prevent tetanus? So that would actually be our plateau phase, which I mentioned earlier. And if you recall from Dr. Cho's notes, one of the slides says that cardiac cells have a special adaptation. Among them are these plateau phase that drag out the action potential. And the point of that is to allow the heart to completely relax before another action potential can start. Basically, imagine your atria need to contract first and then they relax before your ventricles contract. You need to have all of your cells contract one after the other. Otherwise, it might, you know, just be a stiff heart. So, but anyways, so 17 would be A for this funny sodium channels there. And then 18 would be this plateau phase, dragging out our action potential to prevent the heart from just staying contracted. And then more specifically, this is where your pacemaker cells live. They live in the SA node. It's right in that region there. And obviously that's your AV node. And then this action potential travels through your gap junctions in your cardiac muscle cells to reach the bundle and then the Purkinje fibers on the side there. And that's what I just mentioned there. And so now, if you're still having trouble with this figure here, this should sum it up really nicely. So up until now, before this exam, we have learned action potentials in neurons. Neurons are a different type of cell, right? The heart has muscle cells that can behave similar to neurons in that they can transmit these action potentials and they can create their own. So the reason why um, if we look at this graph here, we can see that we're using calcium to cause our cell to depolarize. But if you remember, in a regular neuron, you would use sodium. You have sodium channels that open here. And so that is what explains the difference. We are in muscle cells. This action potential is occurring in a muscle cell. So therefore, we're actually going to be using calcium from the endoplasmic reticulum. So that's why we're using calcium here. But what doesn't change is when we're going through the falling phase, potassium exits the cell. So that stays the same there. And so like I was saying earlier, these muscle cells, they're similar to neurons, but they have certain differences, such as this leaky sodium channel here. And I mentioned this calcium, it's different from sodium here. And then lastly, our plateau phase occurs in these muscle cells. Okay, so that's just helps you separate neurons from these muscle cells and why they're different. So I hope that helps. And then someone said, or this question was, which channels are open at which points in the pacemaker cells? So the only important thing you need to know is that at this phase here, when you're below threshold, your leaky channels are always open. That's what allows it to slowly climb. But other than that, you're still going to have a voltage gated calcium channel here. Once it reaches the maximum, it's going to close, and then you're going to have your voltage gated potassium channels open here. So everything is the same except for the leaky channel we see in this phase and the voltage gated calcium channel we see in this phase. So everything is the same to a neuron basically, except these differences here. And so we'll go over figure 2 and 3 now, but so we're first looking at a muscle belly. This fiber here is a fascicle. You don't need to know that, but that's just what it's called if you've taken anatomy. This fascicle is wrapped by, I think, periosteum, something like that. But anyways, once we zoom in, it's just a group of cells. Once we zoom in, we have our muscle cell. Muscle cell is also the same thing as a muscle fiber. It's also the same thing as a myofiber. So when you see those terms, it's referring to that long cell. This is a single cell. Also note that muscle cells have multiple nuclei. So you will see these dots, multiple of them in a single one of these long tubes. Now, if we look at C, it's pointing at the membrane of the cell, and the membrane conducts action potentials. And I'll get into that in a little bit. And then lastly, inside the cell, we have additional tubes. They're called myofibrils. So hopefully that helps you get oriented. Is B connective tissue? Yeah, B is simply just the connective tissue between your fascicles. And I think it's called peri something from anatomy. I don't remember. But once we zoom in farther, we're looking around here inside the cell. What we see are these myofibrils, and we see these tubes here. These are called T-tubules. And all they are invaginations of the muscle cell's cell membrane. So obviously, we're going to have neurons that reach into the muscle belly, and they're going to put their axons onto our muscle cells here. And then once you transmit acetylcholine, you're going to have depolarization that travels along the cell membrane here. And since we have these T-tubules, which are invaginations of the cell membrane, our action potential will be able to travel down here, deeper into the cell where it can reach all these myofibrils. And then obviously that's what allows, that's what signifies our ER, endoplasmic reticulum, to release calcium. And then you guys probably know calcium binds to troponin, tropomyosin moves out the way, and then power stroke occurs. And that's how you do muscle contraction. And then lastly, we're going to go over how to tell the difference between, someone said, are endoplasmic and sarcoplasmic reticulum interchangeable? Yes, they are. Sarcoplasmic reticulum, sarco just means muscle, so it's the ER of a muscle cell. So first things first, if you see striations, eliminate smooth muscle. Okay, so very simple. Striations are these dark pattern lines. We can see them here as well, but here there's nothing at all. So this would be our smooth muscle. It's very simple. Now, how do we tell the difference between this and this? Well, it's simple as well. Cardiac muscles, remember, they need to share the action potential with the rest of the heart. And how do we do that? We have intercalated discs here. So this is one muscle cell and it's attached to another muscle cell through an intercalated disc. This disc contains gap junctions, and that's what allows your action potential to be transmitted across the heart. And so it's simple as that. This would simply be your skeletal muscle. This would be your cardiac muscle. You can tell because we have intercalated discs, which contain gap junctions. And lastly, this is our smooth muscle because we do not have any of these striations. We don't have those perfectly organized sarcomeres. And remember, one of the reasons why is because we don't have troponin or tropomyosin in our smooth muscles. So I hope that helped as well. And then now we'll move on to the next section here.
I'll share my screen for this.
All right.
Okay. um, Okay, I'll give you guys like a little bit more time to do these. Because number 20 kind of requires some math, but yeah. Okay, I'll give you guys like 15 more seconds. Okay. Okay, so it seems like the majority of you guys got the right answers for every question. Okay. Okay, so 19 is D, 20 is B, and 21 is also B. Okay, so with 19, some of you answered A, and it might be because like technically when if you look at this figure, it kind of resembles like the arterial capillary vein system. And in that system, the blood flow rate is the same across the entire system, but in the capillaries, in this figure, since it's like split into 3 tubes, it's going to change. It's just the overall flow rate is like the overall amount of blood is the same as what's going to be in A and E. But this is mostly like a physics question. So for 19, if you look at B and D, D has a greater resistance compared to B. It's actually double the resistance of B. So if you're going from D to B, you're decreasing the resistance by 1/2. But if you're decreasing resistance, you're increasing the flow rate because resistance and flow rate are inversely proportional to each other. And that kind of has like, it makes like kind of logical sense because if there's more resistance, it's going to be harder for the blood to flow through. So if you think of it as like resistance and blood rate are inversely proportional to each other, then you should get that if you're decreasing the resistance by 1/2, you're increasing the flow rate by 2, or you're doubling it. So if D has 100 milliliters passing through, then B should have 200 milliliters passing through. For 20, this is more like a math equation where you need to find the total resistance. And when it comes to figures like these, you need to try to get it as simple as possible. And the simplest way is to get every section in series with each other. So since B, C, and D are parallel to each other, yeah. So since B, C, and E are parallel to each other, you would try to condense them into one resistor first. And when resistances are parallel to each other, you have to add the reciprocals. So I have it down here. You would do 1/1 plus 1/4 plus 1/2. And then after you add that over, you take the inverse of that to find the resistance of the parallel circuits. So in this case, the resistance of B, C, and D altogether is 0.57. And once you have that, you can have all of your resistors in series. And then it's just like when your resistances are in series, then you can add them all together. And you should get 4.57, which corresponds to B. For 21, this kind of also goes back to the idea of blood flow rate being inversely proportional to resistance. So since B has the smallest resistance value of 1, it's going to have the most blood flow rate because it's easier for the blood to flow through it. There's not much like resisting its flow, so it would be the most active. So just remember like that relationship, that resistance and blood flow are inversely proportional to each other. Conversely, you could say like uh, tube C would be the least active because it has the highest resistance when you're comparing B, C, and D. Also, you guys learned about that equation for flow rate, which is Q equals difference in pressure over resistance. When you're doing those equations, and if you were to apply it to a figure like this, you would have to find the total resistance first, the total resistance of the entire system first. And in this case, it would be 4.57. And that's the value you would plug into that equation to get the overall flow rate, because that equation just tells you the overall flow rate. Okay. So yeah, we'll move on to the next questions. And someone asked if we're allowed to have calculators. I'm pretty sure we are allowed to. At least I remember during my exam, he allowed us to use the calculator. This one is like a bit longer to answer because you have to do a lot of math for these. So I'll give you enough time. I'll give you like 2 minutes probably. Okay, okay, we'll end the poll now, and most of you chose the correct answers. Okay, so the answers to 22 is A and 24 is D. So yeah, these were kind of very heavily math related questions. So for these questions, it's mostly like you need to know those equations that I have listed up on here. And it's mostly like just plugging in, but with the when it asks for like relative change in map, it's whenever you see the word relative, it's usually asking for a ratio. So you basically just need to find the mean arterial pressure for exercise and rest using the values from the table. And then you can find the ratio. Since it says exercise relative to rest, that means they want exercise on top on the numerator and rest on the denominator. And so once you plug in all those values, you should get 1.01, which is the answer to 22. And then for 24, this one is a little bit of knowing that what TPR, which is like the total resistance, you should understand its relationship to the radius of a vessel. So it has an inverse relationship to the fourth of the radius. So basically for this one, you would do kind of like a similar thing to question 22 where you just plug in values, find the TPR for exercise and rest, and then you just need to do the ratio for that and you will get 0.317. But since the question's asking about the change in relative change in radius, you would need to apply it to that inverse relationship that TPR has with radius. And so I have it written down here, TPR is inversely proportional to 1 over r to the fourth power. And that also means that r to the fourth power is inversely like proportional to 1 over t PR. And basically you just need to find like the fourth root of 1 over 0.317 to get the average relative change in radius. But yeah, this one is like mostly, these questions are mostly plugging it in, but for 24 you just need to know that relationship between resistance and radius to get the answer. And for the question in the chat, let me just go back to that. Oh. So yeah, if you were trying to find the overall flow rate for this question, you would just need to subtract the pressures on these sides. You could do like you could make this one pressure 1 and this one pressure 2, and then you could just do 300 divided by 300 minus 285, and then you would have like the pressure differential for this question would be 15, and then you would divide it by the overall resistance, which would be 0.57. So yeah, just to find the difference in pressure, you just need to subtract the pressures on like the left and right side. Okay, I'll give you Maybe 1 minute for this question. Okay, 10 more seconds. Okay, so the most of you put E for 25, and you were between C and D for 26. Okay. So the answer to 25 is E, and 26 should be D. So for 25, when it asks for the volume of blood transported per minute, that is the blood flow rate. And you should just like remember the blood flow rate is the same across the entire system, the circulatory system, whatever you're putting in is the amount that's exiting out. So if you have like 100 liters coming in, 100 liters in the arteries, there's going to be 100 liters of blood also entering the capillaries and exiting the capillaries. So the volume of blood transported per minute never changes. So that's why for E, the line is just going to be like a straight, constant, flat line. But for 26, so when it comes to distance traveled by blood per minute, think of it as distance per time, and distance per time is velocity. So the question's asking about which line represents blood velocity. And when you look at when you look at the system, the blood velocity should be the slowest in the capillaries, and that's because your cross-sectional area increases in the capillaries. So if you look at this figure here, you can see like the amount of blood that enters the capillaries, this amount of blood is the same as this amount of blood. But if you look at the widths of the amount of blood that's moving in these, it's much shorter in the capillaries, the width. And that width is like the amount that the blood is moving by, the distance it moves by. So it is moving like a lot slower in the capillaries. And then conversely, you would say that blood moves the fastest in the arteries and the veins. It moves the slowest in the capillaries because once this same amount of blood gets into the capillaries, it has way more space to move. And that's why it kind of spreads across the capillary bed into this and is compressed linearly. But that is also why it starts moving slower. But yeah, so you should just remember that the arteries and the veins are where blood velocity is going to be the highest, and the capillaries are where blood velocity is going to be the lowest. So that's why D is the answer here. I think the question in the chat, while that is true, there's kind of like a negative pressure that's pulling blood through the veins up back to the heart. I wouldn't say like the velocity is the exact same in the arteries and the veins, but it is like the highest. They're like pretty similar. I don't know the numbers off the top of my head, but it's just the general trend is that it's highest in the arteries and the veins because they have the smallest cross-sectional areas. And that's like forcing a lot more blood to go through in a shorter amount, like a smaller amount of space. So it would move faster. Okay, and then someone had a question about question 20 on topic 17. What would happen to diastolic pressure if artery elastic fibers were removed? And so the correct answer was that it would decrease to near 0 mmHg. And there were also a lot of people kind of asking about the difference between systole and diastole. So basically, systole is when the ventricles, they contract. So when the ventricles are contracting, blood is pumping out of the ventricles through the aorta to the rest of the body and through the pulmonary artery to the lungs. So that's systole. But during diastole, your ventricles are relaxing, so blood is flowing out of your left and right atriums into the ventricles. And that allows them to fill up. But when diastole is occurring, you're not having blood pumped out to the rest of the body. Your aortic valve is usually closed during diastole. So when systole is happening and your heart squeezes the blood out of the heart towards the rest of the body, your pressure is going to go up. But when your heart relaxes during diastole, your pressure goes back down. And it's because there's not like any blood being pumped to the rest of your body. So if your pressure, but like when your pressure goes back down, technically you wouldn't have any pressure during diastole, but we know that's not true, or else we wouldn't be alive. But basically blood pressure sort of goes back and forth between like going up and down until the elastic fibers in your blood vessel, they absorb some of the pressure and they even it out. So when systole is happening, blood is being pumped through your arteries. Your elastic fibers are able to store elastic energy during systole. And then when diastole happens, there's not like anything that's there to maintain the pressure. So your elastic fibers can use the elastic energy they store to exert work on the blood during diastole so that you can maintain a high blood pressure. And that's why like diastole, for systole diastole, when they measure it, it's like 120 over like 75 or 80, because diastole is like the pressure does decrease, but you need to maintain a pressure anyways. So that's kind of like what the elastic fibers in your arterial walls are doing. They're maintaining the pressure by using elastic energy to exert work on the blood. And so that's why they're not there, if the elastic fibers in your arterial walls were to be removed, there's nothing left to maintain a high blood pressure, so your blood pressure would drop close to zero. And yeah.
All
Does
right, should
anyone-
I share my screen or are
Yeah.
there any questions? If not, I will share my screen. All right, can everyone see that fine? Hopefully. All right, so I'll be covering the second half of this cardio topic. Starting off with this question. Which of the following applied to a heart would most likely cause a shift from trace A to trace D in figure 9? So I will give you guys about a minute or so to solve this. All right, probably like 10 more seconds. So for this one, the correct answer is A. I saw a lot of you, or like about half of you, choose D, cure at the SA node. The first thing that we should really start off with is analyzing the difference between trace A and trace D. Really, the big difference here is the frequency at which these impulses, these heart cycles, are actually being conducted or being fired. Here in trace D, nothing here is actually missing, like the P wave, none of it is missing. The QRS complex, none of that is missing. So that means that the SA node is functioning properly in terms of firing these electrical impulses and essentially initiating your heart rate. And as I said earlier, the only thing that is changed is the frequency at which these impulses are being fired. And when I think of the effects of the heart rate and the frequency, I'm thinking regulation by either the sympathetic or the parasympathetic nervous system. In this case, for trace D, the heart rate is a lot slower. So I'm going to think probably some sort of parasympathetic control, either by the release of acetylcholine that binds to these muscarinic receptors on the SA node, or I'm going to think of some blockage of control by the sympathetic nervous system. And remember what the sympathetic nervous system does, it initiates the fight or flight, so it increases your heart rate. So in this case, for these answer choices, there's no parasympathetic answer choice that mentions like acetylcholine. So the answer choice that I'm leaning towards to is actually the beta receptor blockers, because if you inhibit norepinephrine from binding to these beta receptor blockers, you inhibit the norepinephrine's ability to stimulate the SA node in firing the action potentials a lot quicker. So therefore, the heart rate will end up being a lot slower, as shown by trace D. It is not answer choice B, tetrodotoxin, because when I think of tetrodotoxin, I think of inhibition of sodium transmission or inhibition of the firing of action potentials. And if you can't fire action potentials at the SA node or at the atria, essentially you cannot initiate your heart rate. And so typically I would expect a flat line, as shown on these tracings. It's not going to be norepinephrine, because if it's norepinephrine, that's going to stimulate or increase your heart rate, which is not shown by trace D. And it's not going to be Curar at SA node because Curar, what it affects are nicotinic receptors. And in this case, the receptors located on the heart, there are no nicotinic receptors. It's only beta receptors and muscarinic receptors. So if there's no nicotinic receptors, I would expect the tracings to just be normal, like A. So that's why the answer choice is not D and it's A. Moving on to the next question. That okay. Well, then you just saw the answer anyways. But for 28, figure 9, which of the following conditions would most likely cause trace C? I mean, if you saw the answer already, I might as well just explain it. It's D. For this case, answer choice is D. Which of the following conditions would most likely cause trace C? So in this case, for trace C, unlike trace D, here there are these little bumps right here, right? So what these bumps indicate are, they're known as P waves, right? So P waves are basically, they indicate atrial depolarization. So what that means is that the SA node is properly firing and essentially firing these action potentials and allowing this to collect at the AV node. But what's missing in essentially trace C are these QRS complexes. So here there are QRS complexes here, but typically what follows the P waves is a QRS complex. Here it's missing. So when I see that this QRS complex is missing, what that means is that ventricular depolarization is not properly occurring, right? So there's probably some sort of blockage in terms of conduction from this AV node and into the ventricles, right? And essentially, if these electrical impulses cannot spread all the way to the ventricles, these ventricles are not going to depolarize. And if they don't depolarize, they're not going to contract. So for this case, the answer choice would be D, because the bundle of His cells is what actually mediates the transmission of signals or the electrical impulses from the AV node and into the ventricles, right? And if there's some sort of blockage, this transmission cannot occur and the ventricles cannot depolarize, right? In this case, it would not be answer choice C, complete cutting of the bundle of His, because if you completely cut off, there would be completely no transmission to the ventricle myocytes. So that would mean there would be no QRS complexes on the tracing. But here there are some available. So some partial blockage would allow some QRS complexes to be fired, but not all the time, right? And in terms of answer choice B, tetrodotoxin applied to the sympathetic axons leading to the heart. Tetrodotoxin, in this case, would only inhibit sympathetic control. So therefore, you would not be able to increase the heart rate for this. In terms of muscarine at the SA nodes, muscarine activates the muscarinic receptors, and muscarinic receptors are tied to the parasympathetic control. So if this would not match trace C, because muscarine would indicate parasympathetic control, so therefore a decrease in heart rate, which is not shown by trace C. It's just a blockage of the bundle of His, so preventing proper electrical transmission to the ventricles. Okay, hopefully this doesn't. Okay. Now for this one, you guys can participate in this question. 29 is asking how many capillary beds an RBC passes through as it travels from a vein in your right leg to an arteriole in your left lung. And then you could also answer question 30 as well. I'll give you guys around maybe a minute for this one. These aren't too long. Probably another 30 more seconds. Answers are changing frequently. That's right. Let me answer this. Okay, I think I'll go on to the answers for first. Let's start with 29. Um, correct answer is actually A. A lot of you chose B, one capillary bed. But the big thing that you guys should really remember is that when blood is being transferred directly to your heart, there are no capillary beds in the heart. Typically, capillary beds are going to be located at your lungs or the sites where the tissue sites where the blood is being delivered to. So it could be like the liver, your kidneys, your legs, any other sort of tissues or organs. In this case, how I would approach this problem is looking at the direction of blood flow. So in this case, the blood is starting at the vein in your right leg. So when if the blood is coming from the vein, it's going to go directly back towards your heart. So in this case, it's going to travel up into your inferior vena cava and in through your right atria and into your right ventricle and eventually back into your pulmonary artery. So essentially, it's carrying this deoxygenated blood towards your lungs, right? However, um, it stops at the arterial of your left lung. It never really approaches the capillary bed of the lungs, right? So that's what this question is asking. It's asking how many capillary beds actually gets to the arterial, not the capillary bed in your left lung. If it changed the capillary bed in your left lung, then obviously that would be one. But in this case, following this path of blood flow, it actually passes 0 capillary beds, right? So blood is just going directly back to your heart and into the artery of your left lung. So 0 capillary beds. Okay, moving on to question 30. For this explanation, how many heart valves does an RBC pass through? A lot of you chose C, which was, oh, what the heck? What happened there? Okay. Yeah, a lot of you chose C for this question, and that is indeed correct. So basically, as I said, how I would approach this is by looking at the direction of blood flow. So the blood here is starting at the pulmonary artery, right? So at the pulmonary artery, you have this deoxygenated blood, and this deoxygenated blood then travels to your lungs, right? And then the blood gets oxygenated in your lungs, and it travels back to your heart through the pulmonary vein and into your left atria, right? So now pulmonary vein, left atria. So from here, the blood travels through the left ventricle and it passes this mitral valve. So that's one valve, that's your first valve. And then this oxygenated blood gets sent out into your circulation through the aorta by passing through this aortic valve. Oh, sorry, someone's audio is on. Sorry. Okay, yeah, so it passes on through this aortic valve, which is your second valve, and then it goes out into your circulation and eventually through your arteries and into the capillaries in your hypothalamus. So how many valves does it actually pass through? This is the first one, mitral valve, and second one, aortic valve. So in this case, it's 2. Moving on to the next question. It's asking during which segments of figure 13 is blood flowing into the left ventricle? So I will give you guys probably a minute for this one as well. You guys, maybe another 30 seconds. So moving on to the answer for this question. The answer is actually D, which is E and A only. The way I would actually approach this question, it's asking for which segments does blood flow into the left ventricle. The easiest way I would do it is by looking at this blood pressure chart right here. One thing that you should really know is that blood, just like any other fluid, will want to flow from areas of higher pressure to lower pressure. So if blood is flowing into the left ventricle, that means that the atrial pressure has to be higher than the ventricular pressure. So by looking at this chart, which segments does this correspond to? So according to this blue circle right here, we can see that at this section A, the atrial pressure actually exceeds the ventricular pressure. So that's when the AV valve, this atrioventricular valve, will open. And so this will allow blood to flow from the atria and into the ventricles. And here in the second segment, the atrial pressure actually exceeds the ventricular pressure once again at the end of around E, I believe. Or is that E? Yeah. But anyways, at this section, this is where the atrial pressure will also exceed ventricular pressure. And that's when, as I said, blood will flow from the atria and into the ventricles. But then when the ventricle pressure starts to exceed the atrial pressure, those valves will close to prevent the backflow of blood from the ventricles back into the atria. So that's why this answer really is just innate. Moving on to the last question. During which segment of figure 13 is the second heart sound dub heard? So I'll give you guys maybe a minute for this one as well. It'll be another 10 seconds or 15 seconds. All right, so I'll probably move on to the answer choices by now. So the answer to this is during which segment is the second heart sound dub heard? It is in fact C, as the majority of you chose. So at the end of C, or should I start with, the dub sound is usually heard when the aortic valve closes, right? And aortic valve usually closes when the aortic blood pressure exceeds the ventricular blood pressure. Because remember, as I mentioned earlier, blood wants to flow from areas of higher pressure to areas of lower pressure. And typically you don't want blood to be flowing back from the aorta into the ventricles because you just want the blood to be sent out into your systemic circulation, right? To your tissues, right? So at the moment the aortic blood pressure, as indicated by this red circle, exceeds the ventricular blood pressure, that's when the that dub sound is heard, or when that aortic valve closes, right? In terms of explaining when the aortic valve opens, it occurs when the ventricular blood pressure exceeds the aortic blood pressure. So if the ventricular blood pressure exceeds the aortic blood pressure, they will open and allow blood to flow from that left ventricle and into the aorta. I think someone asked in the chat, what effects would muscarine at the SA nodes have? So muscarine activates muscarinic receptors. And if you guys remember what muscarinic receptors, they're associated with that parasympathetic control. So essentially it would just facilitate or help binding of acetylcholine to that muscarinic receptor. So essentially it makes it easier to regulate a slower heart rate. So in this case, as Suba mentioned, it would decrease the rate of firing at the SA node. That's probably the best explanation for that question. But besides that, I think that concludes our questions. So stop sharing. When is someone asked, sorry, when is the lub heard? So the lub is heard when the I think the atrioventricular valves open. So essentially when the atrial blood pressure exceeds the ventricular blood pressure. So if it exceeds the atrial blood pressure exceeds the ventricular blood pressure, that means the blood is going to want to flow from the atria into the ventricle. So that's typically when the lub is heard. I think that's at the end of section A. But yeah.
All right. Does anyone have any other questions? At the end of this, we will upload the video and we'll also upload the PDF if you want to look over any of the questions again.
Thank you, I appreciate it.
No problem. Good luck on the exam tomorrow.
You guys will do great. No worries. People here. Janae? Janae? It's still recording. Oops.